wanted to introduce myself. My name is Jonathan Krieger, and I'm the Applications Manager at BSI. And I hope you can hear me. And if you can't, please unmute yourself and say something or type something in the chat. And so what I wanted to do is give you an overview of the PEAKS Xpro release. And the purpose of this overview is really just to highlight what's changed specifically with both studio and online versions of PEAKS Xpro, and highlight sort of what's new in that. And I appreciate those of you who filled out the poll. So it looks like majority have not upgraded yet to Xpro. So this is something you're entitled to if you have an active SPS. And certainly be in touch with us if you'd like to upgrade or find out how to upgrade, and we can do that. And so once again, thanks for being here. And if you have any questions, you can either ask at the end, or you can try and see something in the chat, or of course, you can of course unmute yourself. And so I wanted to talk about, or start off by saying that what we've spent a lot of time doing is really combining the algorithm of PEAKS Studio and PEAKS Online into one really good algorithm that has the same deployment and is the same algorithm just deployed on different platforms. So we have the PEAKS Studio and the PEAKS Online version. And so what have we included in this new sort of algorithm, or this new version of PEAKS, is primarily support or better support for iMobility mass spec. And so this includes timsTOF. We've put in full support for FAIMS, which we didn't have before. We've worked hard on HDMSE support. And can do that now, and across all of these, where we've strived to put in a lot of effort is the iMobility dimension, so we can essentially use the fourth dimension in feature detection. I'll talk about that. The other thing we've done is we've really worked hard on improving our spectral library search for DIA data, and this, of course, subsequently with the iMobility in terms of FAIMS DIA, DIA passive from the book of timsTOF, and swath data. And so what we've done with that is improve this library search algorithm. We've included a library viewer, so you can view and edit libraries as well as pull in some third-party libraries. And we've really improved the label-free quantification. And I'll show you some data from that. We've improved, as I just mentioned, the label-free quantification. So this goes along with the 4-dimension featured alignment and detection. And we now use that iMobility dimension to do label-free quantification, as well as improved algorithms for missing values or match between runs, whatever you want to call it. It's called different things. I'm just gonna do that. And then, as I mentioned, the spectral library viewer. And so, as this group knows, there are many approaches for turning LC-MS data into protein identifications, and this can be done by a straight spectral library search with a spectral library. Usually DIA is done this way, but as well as DDA to identify peptides. We can do a sequence database search where we use a protein database and match sequences from coding regions in terms of put them into peptides. And we can do de novo, and this is something where PEAKS has really done a lot of work. I just have something in the middle of my screen. And so what you can see is the de novo can really then identify non-coding regions, as well as variant regions and splicing regions. And so one of the things that Peaks does is this called feature detection. And so when you first pull in your data in Studio, it's the data refinement node, or in online, it happens automatically. But what we essentially do is look for an isotopic distribution of a peptide, generally by the carbon isotopes, and then identify this feature. What we've done is now improve this feature detection to both allow a fourth dimension in the ion mobility, so we can look both the ion mobility across M/Z as well as retention time across M/Z. And we've also improved so that we can look at overlapping features, in particularly chimeric scans. And so what does chimeric scans and overlapping features entail, and how does that benefit? Well, if we look at a particular feature that comes off in the same MS1, in this example, it's 823.35 at 30 minutes, and this is just three-dimensional, no ion mobility here. What we can see is we really have two features, a more intense and larger red feature and a less intense green feature. And what's happening is because their M/Zs are so close together, they're not getting filtered out by the quad as separate MS2s, or they're not getting isolated. And so what's happening is in the same MS2, we essentially have two peptides that are these features. And because the software has recognized that there should be two features in this MS2 scan, what we can actually do is use our combination of feature detection as well as de novo and database search to go through and actually annotate both the more abundant, in this case, red feature, as well as the lower abundant ion series that turns out to be the green feature. And so this is really helpful. I mean, when you have a busy experiment, or you have something that you have multiple scans coming off at the same time, this can really help in terms of the identifications, and as well as the quantifications of these. So one of the things that we've launching or putting a lot of effort in is the Peaks Online, or Peaks Xpro Online. And so some of you may know, some of you may not know, but the Peaks Online is really designed to handle massive data quantities on large cohort studies. So whether this be clinical or research, or just a lot of large ion mobility fractionated files, when you think about like timsTOF. And it's designed to do this with a low FDR rate, with large data sets. And so the way Peaks Online really works is that the user interacts with the search engine through a client. And this client can either be command line or a web browser. And what happens is the data is imported right into the client and automatically, and you can set up all your parameters in that Peaks client, which then sends it to the processing server. The server can be held either in the cloud, like an Amazon cloud or Google cloud, or it can be held internally on your own high performance clustering computing node center. And essentially all the work happens there. And then the data is then returned to the Peaks client and sort into this interactive viewer where you can look at your data. And I'll show you some slides from that. But what's really nice about Peaks Online is that it can be, the Peaks Online performance is linearly scalable depending on how many threads you put at it. And so we've done a test with 56 different fractionated samples, which equated to 672 180-minute mass spec runs, or 28 million, almost 29 million spectra. And what you can see is that as you increase the number of threads, you decrease linearly the amount of time it takes to do that search. And so relative performance is essentially increased linearly. and these can be added or downgraded as needed, depending on your license. And so what does it look like? Well, what we've done is really taken the algorithm and designed it for these large data quantities on large cohorts. And way we've done that is made it really, really simple to use with predefined workflows. And so these workflows you can create once, they're creatable, they're editable, but once you've created one, you can save that workflow, and at the touch of a click, you can essentially process all your data using that workflow. And then the other thing that we've put into Peaks Online by request is essentially the pulling of sample names directly from what the initial samples were called. And so you can annotate with your delimiter, and so in this case a period, get rid of the .d, and your whole file name will then be pulled over into the sample name. And so the workflows that are in Peaks Online right now include data dependent acquisition for de novo, PeaksDB, which includes Peaks PTM and spider searches, which most of you are probably familiar with, as well as PeaksQ, which is the quantitation. And this, of course, includes Labelfree, SciLac, TMT, iTrack, and so on. And then on the DIA side, we offer a library spectral library search, as well as quantification, Labelfree. And I'll show you some data from that. The other nice thing to really highlight, and this is one of the biggest differences between Peaks Online and Peaks Studio, is that you can modify the parameters at the touch of a button. And essentially, if you click modify parameters on any run, what you will get is you will bring up your workflow page when you can change any parameter. But what the nice thing is that Peaks Online will only repeat what needs to be done or what's changed. So if you change something in the label-free quantification, say you want to change your groupings or your retention time window, we're not going to go back and redo the de novo. We're not going to go back and redo the database search. It's only going to affect the quantification. Furthermore, what's really nice is if you are doing a large cohort and you want to add in a sample at a later date or remove a sample, you can add in that sample at any point. And again, the software is not going to repeat all the de novo and the database integration on all the previous samples. It will basically just do the new sample and integrate it in with the previous results. And so it becomes a lot quicker to reanalyze data or to add in new data sets. And of course, just like Studio, we offer batch exporting, so you can export either at the sample level or at the project level as well. And so one of the things that was really important in developing this is really good FDR and FDR controls. And so what we've put in, and we've transferred this to the Studio version as well, is we now offer peptide FDR in addition to PSM FDR. And then at the protein level, we offer protein grouping FDR as well as protein scores. So that's up to you. But what we do whatever you select here gets integrated into any quantification that you choose. And so we basically have integrated false discovery controls at the identification level, but as well at the quantification level. And so this leads to more accurate quantification and identification. And so one of the things that's, in the past, Online has had a slightly different algorithm than Studio, and so results would be slightly different. So where we've put a lot of effort is to really harmonize these two softwares so that you get identical or almost identical results between them. And so what we did is we took a HeLa set from Carl's paper here and ran it both in Online and Studio, and you can see that at the de novo sequence level, we're 92% overlap, and this increases when we look at peptides and proteins to 98% overlap between the two software options. So I'll show you a bit more about that as I show you some data sets, but one of the bigger things that we're introducing, and I saw from the poll that some people are using FAIMS, is FAIMS support. And so FAIMS is the new Thermo, well, a couple years old now, but Thermo's ion mobility device that can go on to their Orbitrap series or their Tribrid series of instruments. And so what in the past we haven't really supported that, and the new version of Pro now fully supports that by doing a couple of FAIMS specific algorithm features. And so essentially what we do is we group individual CVs together. And so this allows for a better precursor resolution and feature detection. And then multiple feature associations can be happen per MS spectra. And so what this allows us to do is really increase the depth of protein identification and take into the account that we're going to have different ion mobilities. In terms of different LC-MS runs, that alignment now includes that CV value, or the ion mobility dimension, when looking at multiple runs together. And so what happens is the intensity is really calculated as a summation of a particular peptide across different CVs, which makes it a bit more accurate and a bit more reliable in terms of quantification. And specifically to TMT, we can go in and because FAIMS should theoretically reduce the number of chimeric spectra, we can actually exclude these from the MSMS when we have distinct CV values, which can be separated out. And so this leads to better TMT quantification. And so what we've built in is really a view that, just like all the other ion mobility views, you can go in and you can look at your data, and now in your retention time versus MoverZ view, we can put in specific compensation voltage filters depending on what was used in your run. And then I should also mention that you can use multiple compensation voltages per run. There's no need to run one or two per and complete re-inject. You can go put up as many as you feel necessary in your experimental design. And so how does that look? Well, it looks similar to if you've looked at timsTOF data or other ion mobility data, except what happens now in your feature view is you get a CV value. And this CV value is taken from the scan header of the run and is used in the feature detection. And so what you can see is basically you can look at your feature list, and you can sort by CV value if you wanted to, or compare how your peptides are coming off with different CV values. And so for FAIMS, we now offer a complete solution. And this really applies to DDA, DIA, TMT, and direct DIA in Studio, and can include multiple CV values in a single run. And so what does the data look like, and how does it look? Well, for one of our tests, we took a data set from Pierre Thibault's lab, which was one of the lab that originally adopted FAIMS, one of the labs that originally adopted FAIMS, and they actually searched it with, in their publication, with peaks 8.5. And so to achieve that, essentially, data was separated out by CV value in Excalibur and Thermo's data acquisition software, and then searched as a regular individual run, which is not that efficient, but doable. But essentially in this experiment, they took 3 individual replicates of 293 cells and ran them just as standard MSMS runs, or they split this into 3 fractions that were run with different FAIMS values. And what you can see is in their initial experiment that with FAIMS generates a higher number of protein groups. And when we take this data and actually research it with the new version of Peaks or Peaks Xpro, we actually see a large increase in protein identifications compared to the initial search algorithm. And so we can now actually see around a 36% increase with FAIMS and 20% increase without FAIMS. And that's just due to the better feature detection and better identification as the software has evolved since 2018 to now. And so we see this both across proteins with at least 2 unique peptides, as well as just stripped unique peptide sequences. And how does this look in online? We see very similar results as we do to non-FAIMS data, with around 97% overlap both at the peptide and protein level of sequences. Important to FAIMS is CV values and label-free quantification. And so we take this data and run it through the various platforms with or without FAIMS in Studio or online. What we see is a mean CV ratio of around 0.2. And so what we see is reproducible label-free quantification across the various platforms, and regardless of whether FAIMS was employed. And then what we then sought to do is to look how the software reacts to TMT peptides as well as phosphopeptides. And so we took data from Juan Paulo's lab, who also worked with Steve Gigi. And these are basically 3-hour gradients. This is the original publication, and this is the replicate from the publicly available data set. And so what they did is they took 3-hour gradients of phosphor-enriched peptide and ran it along their mass spectrometer in a tribrid mass spectrometer with various modes, whether they did MS3 for TMT, or MS2 without FAIMS, or various levels of FAIMS with 1 CV, 2 CV, 3 CVs. And the conclusion was that 2 to 3 or having multiple CVs really does increase your number of identifications. And as you can see, if we take the data and research it, we get a very similar output to what was seen in the published data set. So we'll set FAIMS aside for a moment and talk about what Pro has done for increased sensitivity and accuracy. And I'll mention at the end, but we will have ion mobility specific webinars. So if FAIMS is something that you're using a lot of and want to learn more about that, then certainly please attend that webinar. And so what we really wanted to do across all data forms is really look and improve the accuracy of label-free quantification. So as ion mobility becomes more and more prevalent in the field, we've now included a 4D feature alignment for the ion mobility data. And so what you're seeing here is data that was from Jürgen Cox's lab, available, that shows a human yeast and E. coli with a known ground truth ratio between 2 samples. And so basically 2 samples were injected and 3 biological replicates for each sample, and then were quantified using either Peaks Studio or Peaks Online. And what you can see is we see a nice quantification result where we have an expected ratio of 1/4 for E. coli, which is over here, 4:1, and then a yeast of 0.5, which is here, and the human of 1:1. And so what we take into account when we do the 4D feature alignment is both a retention time shift, a M/Z shift, as well as an ion mobility shift. And these parameters are up to the user to set. However, usually for mass tolerance, we recommend using whatever you used for the search. eye mobility tolerance will be up to the user. Usually for timsTOF data, 0.05 is a good starting point. And then one of the things we've put into this pro version is this auto detective retention time shift. And so what that really is saying is let the software decide and go through each individual file when it detects its features and detect what the retention time shift tolerance should be. So you'd no longer have to specify a particular number or minute here. So what does the data look like in the feature view? Well, if you look at any particular protein, you can then go in and obviously look at the coverage map of that protein. And if you pick any particular spectra to look at, again, you will see that. But you can also right click and look at that in the peptide view. And in the peptide view, what you'll see is actually the 4D feature alignment, which includes both the eye mobility dimension as well as the retention time versus MoverZ alignment. And so what you can see here is the top represent one set of samples and the bottom one set of replicates, and the features are primarily really the same shape across all of the samples, and we're seeing the isotopic distribution that we would expect. And so highlighted here are essentially the yeast proteins that should be a 1-to-2 ratio from the humans, which are in this volcano plot right here, and the E. coli's are sitting over here at the 8-to-1 ratio. And so what you can see is that you have consistent grouping of the yeast proteins in the first 3 samples downregulated, and the second set of samples upregulated, and this matches to the group profile as we would expect. And so what we've noticed, and in our benchmarking studies, we're able to quantify more proteins more accurately. And this, of course, applies to the same data set searched online. The only difference here is they were, the grouping is reversed. So the yeast now becomes a 2-to-1 over the human. And as you can see by the volcano plot, we see that the human remains 1-to-1 and the E. coli becomes a 1/4-to-1 as the ratio. The other thing I wanted to mention is when we look at total protein groups, if we go back up, we can see that the quantified protein groups is around 5,594 in studio, 5,774 in online. And so again, very similar results between the two deployments or two platforms. One of the areas in both studio and online that we've put a lot of effort in is called ID transfer. And so essentially, if an ID is found in one run, the software will then search for it in the other runs. This is also commonly referred to in the field as match between runs. And what you obtain as a result on the summary page that really has each sample that you run and a percentage of a missing value. And so the missing value percentage is essentially the number of missed MS1 features that weren't identified divided by the total expected features. And so what you can see is before you use match between runs, we're around the 20 to 25% range, which is a typical experiment. And as you turn match between runs on, you see a drop down to the sub 5%. Usually, these are very similar samples that are replicates and well shot. So, we're getting excellent values in a 6 sample experiment where everything is different. That probably sits at around 5 to 8%, but still drastically better than the 30% that you would be missing without match between runs. So as this is an overview, we're not really going into real in-depth of each of these features, but certainly we do have webinars that will discuss it going forward, and some of you have already signed up for those. One of the neat things that we've put in is this Spectral library viewer. And so as DIA and Spectral libraries become more and more the norm for people are becoming more and more popular with these. What we wanted to do was allow users to really look at their libraries and really edit them if needed. And so really, when you think about the library viewer, it's really a tool to look at your library or import libraries. And this can be done from Peaks projects, whether it's X or Xpro. It can be done from Peaks Online projects. We allow for OpenMS or Spectronaut libraries to be pulled in as a text file, and that text file can be editable as well. So really what this allows you to do is to go into Peak Studio, open the library viewer and pull in a library and look at the components of that library. And so we tell you what the raw files that made up that library are. We give you all the peptides, an ion mobility dimension if you are using, or if it is built off ion mobility data, views of the spectra, as well as the ions, as well as peptide details. And then if you wanted to have an overall view of what your library looks like, this is a drop-down list of various features that include precursor M/Z distribution, peptide length, things like that may give you an overall view or overall health assessment of your library. And so what's really nice is that this is available in the viewer. So if you're a Peaks Online customer, you can of course download the Peak Studio viewer and use the library tool in the viewer version of Peak Studio. So what happens and what does data independent acquisition look like with the Pro? So again, this is the same data set that I showed previously, but this time run in DIA passive mode. And again, just a reminder that 1-to-1 human, 1-to-2 yeast, and 4-to-1 E. coli. And what you can actually see is we get a very, very similar result to the data dependent acquisition when doing a library search. When you take that library search and visualize it in Studio, again, just like data dependent data, you get a feature map as well as protein and peptide tab. And if you look at any of those features, you can get a 4D feature view for both the ion mobility dimension, if you have one, in this case we do, the retention time over M/Z, as well as the ion match between the query, which is your data file versus the library file. And that is also provided in a table view. And then what's really nice is we give you the correlation of the MS2 peaks as the intensity over retention time, and how that correlates to the MS1 precursor. And so the MS1 precursor is what we use for quantification of DIA data. And so what's really nice is you can go in and look at your data and view the alignment between MS2 and MS1. And so just further touching on this, because we are feature-based, if you are in any view, you can always come back to the feature view. And so this is data that you can essentially look at, pick any peptide from your protein view, view it a bit better, or you can go into the feature view and click either any of these buttons, protein, show spectra, or show LC-MS. And if you click that, it will pop it up into the protein. If you click the spectra, you'll see it in the data view. And if you see LC-MS, you'll see it in the LC-MS view. And of course, those of you who are familiar with Studio will know that the different nodes or different dots represent different colors. Yellow little dots mean that there's only a de novo for that, whereas a empty blue square suggests a MS was done, and a filled blue square means an identified by DB search. Similar result in data visualization in Peaks Online. When we go to Peaks Online again, what we show you is this fragment XIC of the MS2s and how that aligns with your precursor MS1, as well as the ion match library. and the same data in chart form. And if you actually click on this XIC button here, what you get is just the XIC of the fragment MS2 from your query. And we remove the library search view, so you can interrogate your ID a bit better if you'd like. In terms of quantification and online, we see the same thing as we see in Studio and as we do previously. You know, there's always going to be a bit more variability with DIA, so you see a few more things, but certainly the bulk of all proteins are at their expected ratios in yeast, human, and E. coli. Another example that I wanted to show is label-free quantification with just DIA data, not DIA passive data. And so we have the ABRF Protein Research Group data set, which really has 4 proteins that are spiked in different concentrations. And so what you can see is when you take this data set, it was run in DIA mode, do a library search, but what we find is across all samples, as we'd expect, really nice correlation and nice reproducibility in terms of numbers of both peptides, peptide sequences, and protein groups. When we go in to look at that further, look only at these 4 particular proteins that are spiked into the mix, we find that, you know, with larger proteins, we have more peptides. That's expected in this case, but we do have excellent coverage of all 4 of these proteins searched in the library. And again, an example of a particular peptide. From this data, you can see that your library match well, and you have nice relative percentage identifications between the ions. So Y9 ion's the biggest in the library. It's also the biggest in our query. Y6 is probably the next biggest Y, and so on. And a nice alignment of XICs. When we look at the quantification of these 4 proteins across the groups, you can see that the group, the expected ratio of this is 1 to 4 to 0.02. And when we look at that based on the group labels, you can see that the ratio is in fact 1 to 4 to 0.2, 0.2, which is what we would expect. And again, this is done based on the precursor, and this is an example of what that precursor looks like. So you can see here, this would be the 4 to 1. These would be the 1 or the ones, or the middle group of 50, and this is the low 0.2 group down here. The other thing we wanted to show you is how we benchmark against other third-party software. And so this is DIA data that was searched by Spectral Library. And you can see that compared to the third party, while the majority of data is centered around the overlapping peptides, that Peaks does tend to see more or identify more peptides, at least in Spectral Library search. And we'll have further data about benchmarking in the later webinars that are on specific topics. But just as an overview, we do quite well against third-party software that we've tested, and majority of the peptides in different acquisitions, whether it be Orbitrap or timsTOF or SWAF and QTOF, are in the middle of the overlapping Venn diagrams. And so I want to end there given the time, but you know, I wanted to leave some time for questions if there are any. I will ask that if you have any questions, to please unmute yourself or type them into the chat box, and I'll do my best to answer them. And of course, you can always, if you'd like to speak to an app scientist or an account manager with specific questions, we're happy to do that or set up that meeting for you. And we also did want to highlight that as we roll out Xpro, we're going to be increasing content on our website, and we'll provide some demo projects there, as well as application notes, etc. for you. And if you have any questions, please contact us. And thank you for your time and attending today.